It's called nearly 1,800 pounds of ready-to-eat salad with chicken products because corn ingredients may contaminate salmonella and listeria. That will just really ruin your weekend. And if you're old, it might kill you. So what I did put in here is this is one that's in New Mexico and Texas. That covers us no matter how you look at it. Either part of New Mexico, part of Texas. Salmonella can cause diarrhea, bloody cramps, and fever. Listeriosis can cause diarrhea, fever, loss of breath, headaches, stiff neck, confusion, loss of balance, convulsions, meningitis, coma, and death if you're susceptible to it. All right, instead of right here, I thought was interesting. Everybody see this one that comes up? And I wouldn't go out and freak out about this one just yet, but it's kind of interesting. So Herpes has been giving decades of research on striking correlates to Alzheimer's disease risk and infection with herpes simplex 1. If you have a specific gene allele. But that could be amazing if this was true, because what would it be able to do? What would you be able to do?
Cure it?
I'd be able to start working a way to cure this and go after it more. Get rid of this.
I thought that Alzheimer's had to do with the protein buildup or something.
It is. They're called beta amyloid plaques that build up in your neurons, but they're not exactly sure what the mechanism is. You also have this other one called tau, which also starts to get all kind of strange in there. But they're not sure. They're not sure the underlying pathogenesis of it. So anyway, I thought those would be interesting. Anybody here? How many people here have been exposed to herpes simplex 1? Just do you think? How many people have been exposed to it?
Doesn't everybody get
Pretty
exposed
much, yeah,
exposed to it?
like 98% of the population. Most people are able to control the virus, or at least clear it. All right, let's start going through viruses, viroids, and prions. We'll have that test and get off to another 2 weeks. All right, viruses are what? Are they the same as bacteria?
No.
Actually pretty easy. So minuscule, acellular, which means what? Where's everybody? Everybody take the day off?
Everybody was afraid of the test.
Yeah.
Are we not going to do the test?
We're gonna do it. We're gonna do it in lab. I sent out an email earlier this morning.
No, I see that, yeah.
Oh, you did? Okay, yeah.
I was like,
So
well,
you,
the testing center.
yeah, you're fine. Yeah, yeah. I'll move it up there then. Okay. I just finished putting it together last night.
Okay.
All right.
So we can go over it today, though, right?
Well,
So
go.
we get an A on the test.
Yeah.
It's like go to chapter 13, we should review.
We'll get through some of this, and you guys got a couple questions at the end. We can go over it.
Okay.
But we got to get through this. Infectious agent having either DNA or RNA cause infections in humans, animals, plants, bacteria, protozoans, fungi, and they've even found viruses that infect other viruses.
What?
So they're basically viruses that infect everything. If there's anything out there that's living, it's got a virus that infects it. Cannot have any metabolic pathway on this one. Causes most of the disease that plague the industrialized world. Why are they saying the industrialized world?
Because we have a good control over bacteria.
Yeah. For a long time back in the day, you had things like the plague and cholera. You still have outbreaks and tuberculosis. You still have those. But those, because of antibiotics, are a little bit more controlled, whereas the viral diseases are still pretty bad. Influenza, HIV, SARS, MARS, Marburg, hantavirus. Am I missing anything? Have you seen that new polio-like virus that's spreading? And who's seen that? Who saw that? What did they say about it? Did you read up on it? I didn't get a chance to read up on it.
It's a different strain of polio, but it's like polio symptoms.
It's not really polio though, is it? It's a little bit different, isn't it? Yeah. But it has the same sort of symptoms, right?
Yes.
Yeah, so it's this virus is only hitting kids, right? So far. Now, is it permanent too, or is it temporary?
I didn't see that.
I haven't read it. I'm looking into that, but it's a new one that's coming around. It's hit 24 people? 24?
I think 36.
36 now. All right. Can either grow or respond to the environment, cannot reproduce independently, recruit cells, metabolic pathways to increase their numbers, no cytoplasmic membrane, no cytosol, no organelles, no nothing, right? Have extracellular, intracellular states, depending on what they're like. So are these alive?
They don't know.
Nobody can make them alive. Kind of interesting right now is the general, like I said, the general scientific argument is yes, right?
Mm-hmm.
Now, this book says, I think yes. The general micro book says no. So they're gonna have to, one of these days they're gonna have to decide whether or not they are or not. I'm not gonna worry about it. Okay, viral genome. You guys, we're just gonna go over a brush on this because I want to get to the viruses later. Virus can be classified on the basis of the host they infect. Are you a plant virus? Are you an insect virus? Are you an animal virus? Did I not do that? Not?
Nope.
I just pulled it off the web. I just pulled it off the. No, I believe you. Oh no, this isn't in there. I just put this in this morning. Sorry. I like the way this works. So you guys don't worry about it. You can write this down if you want. Not that big of a deal. But look at different ways you can classify them. Like I said, bacterial viruses are called bacteriophages. A lot of model systems use them. Archaea have their own viruses. Animal viruses, of course, these are extensively studied because they infect who? Us. Plant viruses, other viruses, see, you can read that out. They just found this out. And now they just found the new dreaded, you seen those? Coming out of the tundra that they're excavating up in the North Pole regions. They're called giant viruses now, and they're coming out of these protozoans. You can actually see some of them with a regular light microscope now. But they don't affect humans. That's one thing about it. Viruses are very specific in which organisms they infect. They cannot infect any other type of organism. You cannot come down with a plant virus. Can you get the doggy flu?
Yes.
No. We'll talk about that in a minute. There's actually a doggy flu out there. All right. Extracellular state is called the virion. And all it consists of is a protein capsule, sort of the hexahedral, kind of the classic virus one, surrounding a nucleic acid. Okay, and the nucleic acid and capsid are also called the nucleocapsid. Some of them have a phospholipid envelope. And they do this when they burst out of the cell, they take the envelope with them, they take the membrane from the host cell with them.
Oh.
And this allows for easier recognition and attachment to the membrane. Okay? So the outermost layer provides protection for the genome and recognition sites. And this is what gives viruses their specificity. Okay? The intracellular state is if you remove the capsid, what do you have left?
Nucleic acids.
You just have the genetic material, right?
Mm-hmm.
Called the capsule smear. And some of these will have some proteins attached to this to protect it too. But basically, once it gets inside, it uncoats and exposes its genetic material. Okay. So this is kind of the, like I said, sort of the classic example of a virus. There's not much to them. So what's their only job again?
To reproduce.
To reproduce. All right. So, in terms of viruses, they show more variety in the nature of the genome than other cells. Okay. So how can you classify viruses? Based on the organism they infect, right? This is the second way you classify them. We've not gotten viruses. The furthest they've gotten viruses in terms of taxonomy is family. This family of viruses. They don't even go into species and stuff much. Because they're just not, it doesn't work out that way. So the primary way you categorize them is based on their genetic material. Are they a DNA virus or are they an RNA virus? They can be one but not both. Every other organism has what? Both, right? So if you were to isolate the cause of agent of a new illness in a patient, and you detect both DNA and RNA. Could it be a virus?
No.
Yeah.
Generally, no, there's actually a caveat, you can say yes on a little bit.
Or
Generally,
it could
you
be
can say-
two-
If it's a retrovirus, you can maybe say what.
Oh.
In general, you're gonna say yes. They can be double-stranded DNA or single-stranded DNA, and they can be double-stranded RNA or single-stranded RNA. Can be linear, segmented, singular, or circular, I'm not worried about that as much. But they're much smaller than the genomes of cells and much smaller than the genomes of bacteria. Why? Didn't mean to get you there. Why is it? Think about it, really pretty simple, don't overthink it.
Because I mean, they're only composed of two things, so I wouldn't need a giant space for.
You gotta have the information to make your protein code, any of your receptors, and any of the enzymes you need. That's it. Everything else it steals from the host cell and invades. So this is new too, so nobody kick out.
Uh-oh.
I didn't see this in here, so you can have DNA, which can be single-stranded or double-stranded. You have RNA, which can be single-stranded or double-stranded. Here you can have what? This one I really want you to look at. RNA, single-stranded RNA, but it's a retrovirus and it's going to convert back to what?
DNA?
DNA. Don't worry about the personality here. Just know the 4 types, what are they? 5 types.
Double-stranded DNA, single-stranded DNA, double-stranded RNA, single-stranded RNA.
And then this single-stranded
Retro.
RNA right here, the retrovirus. Okay? Which ones do you think are going to cause the most problems for humans in terms of cancer?
Oh, cancer? I was gonna say retro would be the worst, but
Retros are going to be bad for sure, right? What would be another one you think?
for cancer, I would think DNA.
Huh?
DNA?
What type though?
Double-stranded.
Double-stranded, those also like to integrate, those also can integrate easily into your
They can hide better, right?
DNA. Well, just like get stuck into your DNA and stuff. The retrovirus are bad because they actually have an enzyme that does that, it's called integrates. They actually integrate into your genome. And we'll go with that in a minute. So, this is an E. coli. Remember how small the E. coli genome looked next to a human one? That little round thing? So this is a single round E. coli genome. And there's a viral genome, maybe a T4 or T5.
What the heck?
Tiny. They don't need anything. All right. Most viruses infect only particular host cells. Specificity, I just talked about that, right? You can't come down with the doggy flu. Thank God. Due to the affinity for viral surface proteins that are complementary to proteins on the host cell, it's called viral recognition. Okay? You've got to have a receptor on the host cell that's going to interact with a ligand on the virus so they can grab a hold of it and get inside. Okay? Maybe so try that a third time. Specific, they only infect a particular kind of cell or a particular host. Can you give me an example of one that is incredibly specific? HIV, it is, it's soap, but it can get chimpanzees, right? Another monkey, it's pretty specific, and it can get in macrophages and some other ones, but it's pretty specific. It doesn't have much of a reservoir, that's why it's been hard. That one is even more so. I'm talking host specificity and cellular tissue specificity.
TB maybe? TB?
TB bacteria.
Oh,
Can anyone? I'm sorry, people always forget this one. They never seem to get it. Don't worry. Only smallpox only affected humans, right? So that one's gone. Polio only affects humans. That's the next one they're trying to get rid of. Affects nerves, tissue, a couple other things. What is one that only infects a particular cell type? And only in humans? How about hepatitis C? Hepatitis C only infects humans, and it only infects what cells?
liver?
Hepatocytes. That's why it took so long to get any drugs or anything for it. It was incredibly hard. They couldn't even grow it in cell culture until probably maybe 10 years now. But it took them forever to even grow this stuff in culture. Generalists may infect many kinds of cells and many different hosts. What's generalist one?
Flu, the influenza, or
Flu, but flu's pretty, yeah, it's kind of a general, actually infects fowls and pigs, yeah.
yeah.
But generally it's respiratory, you can throw flu in there. What's another one? 'Cause these are harder to contain, right? The more generous they are, the harder it's gonna be to get rid of them because you've got more what? You've got more reservoir hosts for it, right? So what's one that's really, that can infect, as far as I know, it can infect any warm-blooded animal. Don't hold me to the fire on this one, I think it can infect any warm-blooded animal. Pretty sure it can. We have it around here.
Oh, wisteria?
Hmm?
Wisteria, right?
Wisteria?
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 Mosquito,
Oh, West Nile?
West Nile.
West Nile can affect a few things. It's got a vector, which is the insect, and
I
it's
know
got,
it affects horses.
and it can, yeah, because there's eastern equine encephalitis too. Those are kind of generalists too. So it kind of depends, right, on some of these. Someone's gonna be more of an owl. How about rabies?
Oh.
Rabies can affect what? As far as I know, it can affect any warm-blooded animal in the nervous system. All right. So this is going to be a tobacco mosaic virus. This is a bacteria that's pretty much what? Had it. Little bacterial pods all over the outside of it. This is going to be HIV by now, probably. Right here. In fact, have you ever seen different flowers that have those weird kind of like mixed colors in them? They almost look like they were liquid and they were in paint. Those are usually done by viruses, actually. They put those in there. There's a virus showing certain different genes inside of them. That just reminded me of that when I saw that right there. Okay, this is general size. So here's a red blood cell. There's an E. coli. This is a smallpox virus. And in the world of viruses, smallpox is huge. That's why they try to use it for lots of gene therapy, because they can pack it full of genes. They need to. What is tobacco? Tobacco mosaic virus. It infects smokers, right?
Mm.
Right?
Yes.
They do infect smokers? People, come on. Tobacco mosaic virus infects tobacco plants. It was a huge, big tobacco blight back in the day. It was cost tons and tons and tons of money back in the day. This is a T4 phage. This is a bacterial phage MS2, and this is poliovirus. Look at the size of these compared to bacterial ribosomes. This little thing can do what to a person? Pretty much kill them. Pretty scary, I think.
What do you mean by phage? I've noticed that the word's being used in the book. And
Phage is what they call a virus that affects bacteria. So
oh,
it's either phage or phage, you can say either one.
okay.
All right, the capsid is to provide protection for the nucleic acids inside, and it provides a means of attachment for host cells. It's going to have these ligands or receptors that are going to be compatible for the host cells. It says composed of proteinaceous subunits called capsomeres, and the capsomere will be made of a single or multiple types of proteins. This is the protective layer of the outside. It gives the virus its shape, but it gives it a means of attachment. Okay, so this is where host specificity comes in. And I just threw this one in too to show you this whole capsid smear. So these are little right here. This is going to be the viral RNA, but it's got these little proteins that are going to be attached to it to give it sort of this structure and some more protection and stuff. They look like little wooden shoes. I don't know who draws them.
It's like claws.
Yeah. All right. This is going to be tobacco mosaic virus. This, I think, looks like a rhinovirus or adenovirus or cold virus. This is just showing electron micrograph of the envelopes here and the capsid fibrous layer. What are these over here?
Rabies.
Huh?
Rabies.
Rabies? Oh, did you look in the book?
Yeah.
Did you look in the book? This is very distinct for rabies. It's called a bullet virus. It actually looks like a what?
A bullet.
Looks like the slug in a bullet. This is rabies. Rhabdoviridae. This is a T4 virus, this is a bacteriophage. What did I say about these things? Can they respond to their environment? Do they have muscles? Do they have nerves? Do they have anything? This is pretty elaborate right here, guys.
Yeah.
Thank God it only attacks bacteria. Human virus is actually pretty boring. We'll go through this in a minute. I want to ask you what you think about it. So, viral shapes can either be what? So what are the three ways you can classify viruses?
The shape.
The host they infect, the nucleic acid, their genome, and the other one's going to be the shape. We're not going to go to shape too much because it's just so widespread. Because I like this right here. Helical, that's the ones that look like this, right? Polyhedral, those are the ones that were just kind of drawing. And then I guess they got bored and decided they didn't want to make up any other names. So the rest are complex. So we're not going to worry about the shape too much. Just know the three different ways you can classify them is going to be what? Genome, host specificity, and capsid shape. Okay? And some of them are more, you can start classifying better. We don't need to do that. All right. If you are envelope virus, when you bud out, you do not lyse the cell, you bud out of the cell, and you release, you steal part of the membrane with you. And then you're going to put some of your virus-encoded glycoprotein, these little spikes in it too. But the membrane allows you to do what? What is that envelope going to allow you to do? So I'm a virus.
Wouldn't it kind of confuse the body because you have a part of like the host cell, so then like the rest of the body won't see it like a foreign.
Try and hide from it?
Yeah.
You could think about that, but what if it came from another person?
Won't it give it protection, maybe?
You know, for-
Protection, maybe. What?
Yeah.
Huh? Well, what do you think? What do you think? What do you think might be the advantage? Because usually it makes them more fragile, right? They can't survive outside as long because eventually this thing's going to dry out. They can't survive being exposed to chemicals as long because you damage it. So obviously it's important, right?
Yeah.
Otherwise it wouldn't matter if you got rid of it, right? So what do you think it may allow the cell to do, or the virus to do?
Connect to other cells much easier.
Good. Membranes like to do what with other membranes? They like to fuse. So this gives a nice way for the virus to attach. It can pull down and it can fuse that membrane and it can get phagocytosed or endocytosed right inside. So it makes it a nice easy way for it to get inside of the cell. So envelopes play what?
Attachment properties.
And glycoproteins often play a role in host recognition, but also in host infection. It allows them to get in more easily. All right, so again, here's going to be lipoproteins, just going to be some matrix, like a shell. Here's that capsid, but here's going to be what?
That's one membrane.
That phospholipid membrane that stole when it left, okay? Took with it. HIV likes to do this. Hepatitis,
And its main reason is attachment, not to hide?
it's
I would
no,
think.
yeah, no, because think about it. If I take a cell from you and put it in you, is it going to be able to hide from it? Because it's still going to
Oh.
have some of these little markers on it, right?
Yeah.
The microaggressions. It's not that. It's going to be host recognition and attachment and integration. Because remember, when this thing binds to another membrane, it's going to want to do what right here? This is going to attach the receptor. That'll attach the receptor to pull down to the membrane. And what can it do next?
Oh.
It can
when
fuse
it-
right with that
yeah.
membrane and get drawn right back in.
Yeah, okay.
Makes it a little bit harder. So, again, I just put these properties of viruses in here versus cells. Don't worry about that. You all should know that by now. All right. So viruses classification is going to be what? What do we think?
Nucleic acid.
Nucleic acids, presence or
Envelope.
absence of an envelope,
Shape.
size, shape. What else is not in there?
What it
The
affects?
host type.
Hmm?
What it affects?
Host recognition, yeah, host specificity. Of course, that's going to be based on a couple of these other things too. Remember, virogen have only been organized to the family level, and even then it's sketchy. So the relationship of a virus is not well understood by taxonomists, so you don't even worry about it. We're not even going to do it. They do have them in families, family Viridae, family flavivirus, family, what's HIV, lentiviral, but they're just not that. You don't even worry about it that much. Why could it be important to know family though? Why would it be, listen, you little, get a stinking Monday morning, bright and early. Why would it be important maybe to start getting these evolutionary relationships or taxonomic relationships?
Because if we understand how the family works, we can understand how to stop it.
Good. If you can start getting these families together, you might be able to start designing drugs that can deal with an entire class of viruses as opposed to just individual viruses. All right, good. All right, viral replication, you need to know this. This is the lytic stage. Lytic stage means it infects, gets inside, replicates, and then bursts the cell open and dumps a bunch of virus into its environment. Cold viruses go through the lytic stage. Right? So viral replication usually results in, not usually, as far as I know, goes through the lytic stage and always results in cell death. It's going to lyse the cell open. 5 stage is attachment, which is host recognition. Entry has got to get inside. We talked about getting those new drugs that prevent it from getting inside, right? Right? Synthesis, which means it starts making the parts it needs. Once you've made everything, you start assembly. And then once everything's been put together, you can do what's called release, either bud out or lyse the cell. In this case, it's going to be what? Lysis right here. Okay. I guess some budding viruses actually go through the lytic stage, they don't actually lyse the cell. So here's lytic stage right here, it's the bacteriophages, the T4. Somebody explain to me what's going on there, because I just said viruses can't move. So we'll go down here, and I want to go back up here. Attachment, virus attaches, right? Receptor recognition. Entry, it injects, in this case, bacteriophages usually inject their genome in because they can't get through the cell wall, right? Now the bacterial chromosomes in there, it's going to do what? It's going to start making what it needs. And some of the things it's going to do is going to start chopping up the bacterial DNA, because where is it going to get its DNA from? It's got to get them everything from the cell it's in, right? Okay, now it's got made all the parts it needs. So now you got all these proteins, you got all the synthesis stage, and you go to assembly. And it's just like in a car assembly line, right? Base, tail, sheath, DNA gets packaged in the capsid, add the capsid onto the sheath and the tail, add the tail fibers, and now you've got a what? Mature virus. When they're all done, it's going to lyse the cell, open up, and go back out and do what?
Do it again.
Back to other cells.
What happens to the original one?
It's going to be gone. It's going to be stuck to the top up there, especially on these. It's just going to, it's kind of like a, I don't know what you would call it. Kind of like a little drone thing. Attaches, injects its DNA, and then it's done.
And it
So
just
there's
is.
nothing else inside of there, right?
Right.
How, look at, what is it doing from pain A to pain B? From attachment to entry, what's going on?
It's,
Somebody explain to me how that's happening. From entry here, what's it done? Those little tail
Attached.
fibers are the ones that have host recognition, right?
right.
So they're going to interact with these receptors on the surface of the bacteria. What's going on here?
It's injecting its DNA.
What else does it have to do?
Has to like pierce through the membrane.
Does it look like it grabs a hold and pulls onto it?
Oh, the
Yes.
tail sheath pushes through.
How does it do that?
It has to have some sort of.
Some sort of, that's a sticker right there, some sort of what? How is it gonna do that?
Piercer?
Right? Does it have nerves?
No.
Does it have muscles?
No,
Does it have anything?
it chemically reacts.
How does it do it?
Destroying the
How does it-
peptidoglycan layers.
Huh? The what?
Destroys the peptidoglycan
It
layers.
actually does, a lot of them actually have lysozyme in them. Remember we talked about lysozyme? One of the things it does, it injects lysozyme. Well, look at this little drill bit looking thing it's got. So the scripted lysozyme breaks down the peptidoglycan layer, inserts that tail spike. How is it doing all this? With no
Bruce,
muscles.
is there some kind of pressure working on it?
No, I don't think it could be. It could be a pressure gradient. Maybe you can push the thing down possibly. What's going to change that pressure gradient?
The proteins that are inside of the tail, because everything's made of like proteins, right?
Right.
So then maybe like the plasma membrane doesn't realize that it's like an outside protein. It's just like, oh, it's a protein. Let's attach it to the membrane, and then it.
So it's gonna try and bring it in?
Yeah.
Could be, except you're attaching to a cell wall unless it's a gram-negative bacteria. Although that looks like that might be a gram-negative bacteria. Good, could be the bacteria itself, or the cell itself pulling it in, or what do enzymes do when they change shape? What's that called? Called a conformational change. Remember, proteins can do what?
Change.
Like receptors and transporters, they can change shape, right? So they can get some sort of movement. So when they get here, these tail fibers can maybe do one of two things. Either get drug in, or they can undergo a conformational change and pull it down, and then everything gets back. So they can kind of change shape, no matter where, not move. All right, good. Lysogeny is a modified replicative cycle. Infected host cells grow and reproduce normally for generations before they lysis. They're called temperate phages, so they get in there and hide. And I want you to know this word right here, because you'll see this later on. Called a prophage. We'll talk about proviruses later on, because that's a big deal in HIV. Prophages are the inactive phages. They're just the bacteria, the phage nucleic acid inside of the cell, right? And lysogenic conversion results in phages may carry genes that alter the phenotype of a bacteria. Sometimes the virus will bring something in like a bacterial resistant gene, or something that allows it to use another carbon source, or something, okay? That's lysogenic conversion. But what's going on here? So now you have this lambda virus attaches. In this case, it goes through the lytic stage and just does what?
Reproduce.
Reproduces and kills the bacteria. Every once in a while, it converts to lysogeny. What's going on in lysogeny? What's the advantage?
It makes more opportunities to reproduce, like make.
So what's it doing here?
It's like building factories. Like it's giving them opportunities to make more.
Good. Once you incorporate your DNA, and it always incorporates in these phages. It always gets in the bacterial DNA. Every time the bacteria reproduces, it also copies what?
The virus.
The viral DNA too. So the virus gets to grow and grow and grow without doing what?
Busting it.
Anything. It's just going along for the ride. It's making millions and millions and millions and millions of copies of itself. Then something will happen. It's called induction. The bacteria may start running out of energy. Something may go on. It causes the virus to come out of lysogeny and go back into the lytic cycle. So now it can just reproduce and make billions of copies of itself too. But it's a nice way for the virus just to do what? Just hitch a ride, right?
Yeah.
And not have to do anything. There's a very similar thing in us. Like I said, who's had chicken pox again? And I'll make us raise our hand all the time.
I've had
We have
chicken.
very similar mechanisms inside of us.
Is that how it turns into shingles?
It's basically the same thing, a little bit different because we don't go through this. It's a little bit different than the lysogeny, but it's close. And that's what we're looking at here. Basic replication pathway as bacteriophages. So a lot of them go through the lytic stage. If you've got a cold, what happens? You get infected with a cold virus, it does what? Infects your upper respiratory cells, it just goes to that lytic stage. That's what makes you feel so crummy, right? It lyses the cells, elicits an immune response, you just feel miserable. Okay, differences result from what? The envelope. Good, true, or false question. There are envelope bacterial viruses, or bacteriophages. True or false?
Wait, there are.
There are enveloped bacteriophages. True or false? Pretty much false. There are a few rare exceptions, but in general, it's false. Eukaryotic nature of animal cells, they're more complex. When you get inside of a bacteria, what do you have to work with inside of a bacteria? If you're a virus. You don't got much, do you? You've got the ribosomes, and you got the nucleic acids. That's about it. Inside of eukaryotic cells, you've got what? A whole host of machinery for different more complex proteins, possibly. Lack of a cell wall in animal cells. You don't have to penetrate that cell wall. It's much easier to get inside. So, chemical attraction between viral proteins and cell receptors, and we've gone over this time and time again, because we've talked about how can you develop a drug to protect from a virus. How do we say you can do it? You can what?
Change the shape of it.
Either get a drug or an antibody that's gonna block the receptor. If you block the receptor, can the virus do anything? No. Animal viruses do not have tails or tail fibers or cool little heads or cool little everything else. Those are only bacteriophages. Animal viruses are pretty boring. They have glycoprotein spikes and other attachment molecules that mediate attachment. Talked about HIV. What did I say about HIV? What are the, do you remember the 3 receptors it has to use? It's got to use one for sure. That's why it affects that particular cell type mainly.
Do they like they're the ones that have those glycoproteins?
They have glycoproteins, but the receptors are going to interact with those glycoproteins. So CD4 is going to be one of them, and CCR5 is going to be another one, and CXXR4 is going to be another one. So it has to have those, otherwise it can't get in. All right, know these 3 ways to get inside the cell, guys. This guarantee will be on the desk. If you are a non-involved virus, and all bacteriophages do this too, it's called direct penetration. You attach to the outside of the cell, kind of like a bacteriophage, and what do? You just breach that membrane somehow and inject your nuclear material. The nice thing about this is you have to uncoat once you're inside. So would a drug that prevents uncoating have any effect on this? No. Membrane fusion, we talked about the 2 different reasons for being an envelope virus. If you're an envelope virus, you interact with receptors on the host membrane, your envelope now fuses to that host membrane, just opens up and what? Dumps the virus capsid inside, then it uncoats. The other way is going to be what? Receptor mediated endocytosis. Pretty much the same thing. What's the difference? If I ask compare and
It kept its
contrast.
coat. It kept the membrane in this one.
Right. This one it opens up and just sort of moves through
Dumps
the
it.
membrane. This one's brought in through a phagosome, right? This phagocytosis is brought inside. All right. Synthesis of DNA viruses, double-stranded viruses. If you got double-stranded, what can happen? You get inside, you uncoat your double-stranded DNA, what can you do next?
Attached to the host something.
But the mechanism is pretty much already set up, huh? You got double-stranded DNA, your DNA is double-stranded, all of those hosts, RNA polymerase, and everything else are ready to go. So similar replication in the cellular DNA. The viral genome is replicated in the nucleus, just like your DNA is. It's nice and simple. And the viral proteins are made where? In the cytoplasm. Just like you, right? You get transcription and DNA synthesis, and you go out and get translation.
So it's faster?
Pretty quick. They don't have to do much once they get inside. Of course, we're talking about viruses fast. It's sort of a relative term, but yeah, everything's good to go. If you're a single-stranded DNA virus, we don't have what? Single-stranded DNA. So what is going to happen first?
Has to make another strand?
So the host enzymes produce a DNA strand complementary to it. You're going to go in there, your enzymes are going to see that and say, wait a minute, what's going on here? Why is there single-stranded DNA in here? What is it going to do? You're going to add polymerase to it, you're just going to copy the other side of it. What has that basically just done to the size of the genome of that virus, though?
Double.
Just about double the size of its genome real quick, right? So, now the viral genome is double-stranded, and then you can do what? Go right through transcription and translation and synthesis, same thing. They're pretty easy, the DNA viruses. RNA virus is a little more complicated. So synthesis RNA virus differs from DNA viruses. You're going to have positive sense viral RNA. They can act directly as messenger RNA, which means you can just start reading it right away. Or you have negative sense RNA viruses, which cannot be translated directly. So they don't act like messenger RNA. Okay. So you have single-stranded RNA viruses, positive sense, retroviruses, negative sense, and double-stranded RNA viruses. So we'll look at how that'll work. This is probably the worst slide of the whole thing. Okay. If you are positive sense Single-stranded RNA, what do once you've got
Dump your DNA in there.
translation of our proteins, genome acts as messenger RNA. Bam. Once you uncode, you can start doing what?
Working.
Translating and transcribing. Translating into proteins, right? The only part that holds you back, you've got to do what? If you're just using this for messenger RNA, right, to make all the stuff you need, how are you going to copy your genome? So now you got to take this, go back and do what? You got to make a complementary negative sense RNA that acts as a template, and then you turn around and copy that into what? Positive sense RNA. Now you have what? Copies of the positive sense RNA. This side over here has been making all the parts you need, and then you just assemble and what? If you're a negative sense, what can you do? Or what do you have to do first? You can't act as messenger RNA, so you've got to do two different things. Transcription by RNA-dependent RNA transcriptase, which just means you do what? You convert negative sense to
Positive.
positive sense. Okay? Now you get further transcription, so now you're going to make copies of your genome, so you're going back to the negative sense, right? And on this side, the positive sense can now act as what? Messenger RNA. So now you can start making copies. If you are a double-stranded RNA virus, you unwind, and a negative sense transcription by viral RNA polymerase to make the complementary strand. Again, it's a lot like over here, right? You just take a step out.
Oh.
On this side, the positive sense acts as a template and makes what? The viral protein.
So one is working to copy itself and the other one's already working.
Right. So all you have to think of is what does it have to do first?
Okay.
Okay. If it's positive sense, it can start doing what right away?
It can start.
Positive sense can start acting as messenger RNA right away. It's got to copy the DNA genome. Double-stranded can do both at the same time, right? Negative sense just has that extra step. It's got to do what?
Turn to positive.
It's got to make that into a positive sense. So it can start using that as messenger RNA. Make copies of that. And I'll bring up all the different enzymes. There are RDRPs, which are RNA-dependent RNA polymerases, and there's RNA transcriptases. I may have put them in here. I did not. Retroviruses, on the other hand, do what? What is the central dogma of biology? How can I not get through a chapter of this class ever? What? It goes from RNA to DNA back to RNA, then the protein. So it does not use their genomes as messenger RNA, period. All they use it for is to make what? Double-stranded DNA. So use a DNA intermediary transcribed by viral reverse transcriptase. That's what I want you guys to know about this one. So I put this in this morning too. Show you what happened. And this is why it's so insidious. HIV just infected somebody. What's going to happen? Intra or uncoding of the retrovirus. So now you've got what? Single-stranded RNA, right?
A single?
But what's it got to do first now?
It's gonna make a copy.
Can you use reverse transcriptase to do what? 2 steps. Reverse transcribe this into DNA, and then the DNA is going to be copied into double-stranded DNA.
Okay.
Now it's going to go into the nucleus, and this is the problem with retroviruses. What does it do once it gets in the nucleus?
It integrates with your DNA.
It integrates into your genome. And once it integrates, what did I say happened?
It's changed the DNA.
We can change it, but that's part of it. But what I say in terms of you as a patient, what happens? Once it gets in there, it integrates into your genome.
It starts
Body
making a
will
way.
replicate foreign virus.
Can you get rid of it?
No.
No, you're done, right? So integration means it's in your viral DNA. Your viral DNA is in your DNA. Transcription by host, and now what can it do too? Does it have to make all these viral RNA polymerases?
No.
Or these viral RNA DNases? All I had to do is reverse transcriptase to do what? Copy that into double-stranded DNA eventually. Now what does it use? Transcription by host RNA polymerases forms viral messenger RNA and genome copies. So it completely does what in terms of your cell?
Hijacks.
It completely hijacks it, right? As opposed to the other ones still have to have these things they call RDRPs, RNA transcriptases, all these other things. Once it does this, it's taken over your cell, and it uses everything in your cell just to make copies of itself.
Does your cell release like, I think it's MHCs to show that it's been
Yeah.
affected and then it can be attacked?
It does, yeah.
It
That's,
does do that?
yeah, it does. So your body, so that's actually a good question. With HIV infection, do you elicit an immune response?
Only when your cell gets attacked?
No, yeah, no, you do, you make antibodies, you make T cells to it. What's the problem with HIV?
It attacks your helper T cells.
It's going after your helper T cells, right? Which are the things that orchestrate your entire what?
Immune
Adaptive
system.
immune response. So why does it take 10 years on average to go from HIV infection to full-blown AIDS?
Because it's still attacking your helper T cells and it has to bring that down.
Right, so what's going on over those 10 years is there's a big giant war going on inside your body. Your immune system's going after it as fast as it can, the virus is replicating as fast as it can, eventually who ends up winning?
Virus.
HIV ends up winning eventually, in every case. So, yeah, so that's why it takes so long. You elicit a pretty robust immune response, but it's just,
So
you
if
can't
you
keep
do
going.
react, it just,
Mm-hmm.
Yeah, that's why they have
HIV
cold
is so much
symptoms
more
and flu
rapid.
symptoms, right?
Right, there's a thing called, what's it called? It's called HIV. What is it called? HIV, not complex, HIV, not syndrome. A lot of people, they get infected with HIV about 2 weeks after they get infected initially, they feel like they have the flu,
Mm-hmm.
except without the respiratory stuff. And that's about initially, you initiate that initial immune response against the virus. And it comes on, and it's actually the viral load, if you watch the viral load, it goes up like this, and then drops down.
Mm-hmm.
And then it starts taking over, slowly starts working its way back up. And does that mean, is there probably somebody out there that's been infected and just gotten rid of the virus?
Maybe.
What do y'all think?
I think so.
Do you think so? What do you guys think here?
early stages.
What do you think?
Mm-hmm.
Do you think there's some guy out there who got infected and was able to fight off the virus? Probably. My guess is, yeah, there's probably some people out there that have been able to do it.
That's crazy.
Problem is, once it integrates, it's tough. But
Imagine they didn't even know. They just thought they had the flu in
they
the way.
got
That
it.
probably happens, I think.
Probably has happened, and I'm pretty sure. All right, guys, that's it. So go. Sorry I didn't get to review anything. I'll put that in this form. A little later, so just